Hello, everyone. We'll just give everybody a minute to get settled on here. Just give it a minute or two. Okay, hopefully everybody's all settled, and if not, that's okay. I'll get started on our introduction, and then we can take off. So hello, everyone, and welcome to the Invasive Species Asian Carp Canada webinar series. My name is Jenna White. My pronouns are she, her, and I'm the Policy and Program Development Intern at the Invasive Species Center, where I also work on our Asian Carp Canada program. I'm thrilled to be your moderator today on this beautiful Monday afternoon, speaking on the status of black carp in the United States. So while Invasive Species Center is located in Sault Ste. Marie, Ontario, I'm speaking to you from Sudbury, which is the Robinson Huron Treaty Territory and the traditional homelands of the Atikameksheng Anishinaabe. Having lived here since time immemorial, Indigenous peoples are the original stewards of this land, and I would respectfully acknowledge their long history of community and identity in these areas. As a settler descendant, I'm grateful to have the opportunity to work and live here, and thank all generations of Indigenous people who have taken care of the land before me. So in case you are not familiar, for some context of who we are, the Invasive Species Center is a not-for-profit organization that connects stakeholders, knowledge and technology to prevent the introduction and spread of invasive species that harm Canada's environment, economy, and society. So we've got a number of great resources related to invasive species on our website, including species profiles, best management practices, and more. So come check us out at the invasivespeciescenter.ca afterwards, and you won't be disappointed. Asian Carp Canada is one of several programs under the Invasive Species Center. So ACC was created to convene information on the most recent prevention technologies, early warning measures, response efforts, and the overall threat of Asian carps to the Great Lakes and beyond. The project components aim to enhance education and knowledge of Asian carps, and we do this in partnership with many agencies across Ontario. We have more great resources that you can find on our website, including recording of our webinar series, including this one, species profiles, risk assessment summaries, and we also have social media channels and information sessions as well. And we also have an exhibit at the Toronto Zoo. So before we get started with today's webinar, there's a few things that I was hoping to cover. There will be time for questions at the end of the webinar. So if you have a question at any time, please type it into the question box and I will read them to our presenter afterwards. If you're having any technical difficulties, please type them as well into the question box or respond to the email found in your registration and we will try to resolve them for you. And lastly, there will be a very brief survey following the webinar. So if you could take some time to fill it out, we would really appreciate it. And now for our presentation. I'm pleased to introduce our speaker, Patrick Kroboth. So Patrick Kroboth is a fish biologist with the U.S. Geological Survey, Columbia Environmental Research Center since 2017, with a research emphasis on invasive fish species, specifically invasive carp, and the subject of this talk, black carp. And forgive me, but I will save your ears from the Latin pronunciation of the term. But Patrick has completed his master's degree at Mississippi State University, studying larger habitats of sturgeon, and has a bachelor's degree from Virginia Tech with research on invasive northern snakehead. Thank you so much for joining us today, Patrick. And with that, I'll pass it over to you.
Thank you. Can you confirm that you see the slides?
Yep, looks great. Thanks.
Thank you. Okay, thank you for having me. A little bit of background. This talk was requested as an update on black carp status in the United States and current research, and this is being given in preparation for hopefully a later presentation by Andrew Drake on the binational risk assessment. So with this talk, I plan to not include a few topics, particularly potential invasion pathways, which I anticipate will be covered in that binational risk assessment, and a Great Lakes centric topics such as population or food web modeling. Some things that I do plan to discuss will be work by myself and colleagues throughout with references to individuals. And then just starting off, I thought the best place to go from was, what are black carp? With that, I plan on covering the background. This is kind of an outline here, and I'll follow this outline throughout the talk so we can kind of keep pace. But I wanted to provide some background for you, some history of use, identification methods, and then I'll get into the research and distribution, population parameters, ecological effects, some methods for detection, and then also some methods for control. So black carp are native to large rivers in eastern China and Russia, and you can see the map here on the right-hand side is from Nico et al. 2005, and that's an initial biological synopsis and risk assessment on the species, and that's a very good reference on this species in particular, and I'll mention that throughout the talk several times. I'll bring it up. So if you're interested in kind of the base knowledge on the species, I would recommend you look towards that reference. But again, the native range, large rivers in eastern China and Russia, and you can see them pictured here in blue with the Amur in the north, moving south, the yellow, Yangtze, Pearl, and Red river systems. Black carp have the ability to be fairly large-bodied fish. I believe, hopefully you can see my cursor here, but this fish is the largest that we've had reported in the United States. It's a 1,607 millimeter total length fish, 52 kilograms that was collected from the Middle Mississippi River. Total length, if you're not familiar with fish and fish measurements, is measured from the tip of the snout to the tip of the caudal fin. And I will go over on a later slide the different sections of the Mississippi River Basin and subbasins so that we're all kind of on the same page throughout the rest of the talk. But just know that this is from the Mississippi River. This fish is also from the Mississippi River, similar kind of area, the Middle Mississippi, a 1,300 millimeter total length fish. This fish in the lower left, if you can see my cursor, is a 1,580 millimeter total length fish. It was from the Osage River in the Missouri River system. It's a major tributary, and it was 51 kilograms, again, 1,580 millimeters total length. So this is actually the largest recreational capture that we've had reported. And then I wanted to provide this image for you just to get a general idea of what that length means, because everybody can be different heights, different builds. A picture next to a person kind of varies, but this is a standard tailgate of a pickup truck. And you can see that this fish this is actually from the state of Mississippi, and a biologist sent it to me. This fish is about the length of the length equivalent to the width of a standard tailgate. So just kind of relative size. So you can see that they do have the potential to grow to be fairly large. So large-bodied fish, and with a large-bodied fish, they can have a large mouth. A mouth gape is one of the limitations of a fish's ability to ingest prey. So they also have a special adaptation, these molar-form pharyngeal teeth on their pharyngeal jaw, and this is not the standard jaw teeth that you might think of in the front of the fish's mouth. This is actually an adaptation of the last gill arch. And these teeth are adapted for exerting a fairly strong bite force. And I kind of wanted to show you the orientation here. So they have the image on the upper left is one that we took, and they've been studied for various purposes. This manuscript cited here in these images from Gidmark et al. 2015. This is actually the appropriate orientation. These are the lower gill arches. And you can see this is a cross section from that. Gidmark looked at the functional feeding morphology of black carp by various methods. I wanted to show you that, hopefully you can see this from my scan. These are some of the teeth. This is a 36 kilogram fish, but they're arranged in a 4-5 pattern. And again, the jaws on the bottom, on the roof of the mouth, they have a keratinous pad here that they use to crush the prey against. So that's in that diagram, the PAD acronym. These teeth are also fairly strong. They've been studied as a potential engineering material, and Hay et al., the other image that I have here for the enamel. So we have a combination of a few things. We have a fairly large bodied fish, powerful pharyngeal teeth, and combine that with the potential prey. Many of those species of mollusks are imperiled. So obviously we have a concern for this invasive species. Black carp were imported to the United States in the 1970s and 80s for use in aquaculture. Their primary use was as a biological control of snails in aquaculture. Those snails serve as the intermediate host for digenetic trematodes, and those trematodes can infest aquaculture stocks, for example, catfish or hybrid striped bass stocks, and have an impact on those. Most of the research, if you look back at this or their prior use, has kind of been around the emphasis of black carp as a food fish or for that biological control of snails in aquaculture. Black carp were listed as an injurious species under the Lacey Act in 2007, and the Lacey Act is the United States regulation that prohibits the import or shipment of restricted animals, birds, fish, or plants. Over time, we've seen escapement in the wild with increased reports from the wild, and I'll go over that in some of the later slides, but this is where I wanted to point out, the various subbasins to you. And so starting from the west and moving east in orange, this map is actually from an outreach material, so I'll highlight a few other sections that aren't quite shown on this, but in orange, we have the Missouri River Basin. In purple, we have the upper Mississippi River Basin, which is also divided into the middle Mississippi River Basin, below the confluence of the Missouri down to the confluence of the Ohio. The Ohio River here is shown as the watershed in pink. And then we have the Cumberland River in yellow and the Tennessee River in blue. Below the confluence of the Ohio is the lower Mississippi River, and that runs the length of the Mississippi from the confluence of the Ohio down to the Gulf of Mexico. And there's also some basins over here that aren't highlighted. The Red River here meets with the distributary of the Mississippi and forms the Atchafalaya River. And I'll mention that in later slides of this talk as it runs down to the Gulf of Mexico and terminates. And there's also in the state of Arkansas here, we have the Arkansas and White River systems. And they actually connect fairly closely to distribute into the Mississippi River. So I'm moving on to identification. In working with the species for several years, we've worked with the public, a lot of commercial fishers, and that's the source of a lot of our reports. We've been able to identify some of the commonly confused or misidentified species. As far as native species, blue suckers are the most commonly misidentified or reported. Blue suckers are fairly easy to differentiate. They possess a falcate dorsal fin and a section of the dorsal fin that runs along the length of the back. So this longer section here is always present. And from a fish biologist perspective, they have a different morphology in general, somewhat elongate, also different mouth position, fin position, everything else. But that's an easier method to differentiate when working with the public. We've seen only a few instances of buffalo being misidentified. That's just because of the size of the fish. That really the size of the fish's scales, but they do tend to have a deeper body in general, and they're fairly easily differentiated. We don't get a lot of reports from those. As far as non-native fish, which is the next column I have here, I have black carp on the top. Common carp is another one that comes up in a somewhat regular report, but you usually have a darker colored common carp. They're pretty easy to differentiate based on the presence of a barbel in the corner of the mouth, even though those can vary in size. There's some other morphological methods to differentiate them. But grass carp are definitely the most commonly misidentified species, and that led to us preparing actually a manuscript and some outreach materials to assist with identification. So the most commonly reported size class that we see are what we call large juveniles and adults, because black carp mature at fairly large sizes. In females, it's probably around a meter. Males, about 800 millimeters total length. And the prior keys for grass carp and black carp identification always highlighted the use of pharyngeal teeth and external color. Now, pharyngeal teeth, as I mentioned before, those are fairly dense bones, and they're actually embedded in some of the most dense muscle tissue within the fish's body to be able to exert those bite forces. So there's a significant amount of effort that goes into being able to extract the teeth for identification. And there aren't many in the public or commercial folks that will want to go through that effort to be able to identify a fish. Also, coloration, I mentioned, is a method, but that can vary with capture location. For example, grass carp that have been collected in tannin stained waters are usually commented as being darker in color than say a more turbid system. So we needed a method for quick external identification and a simple character set to help with that. So we went back and looked at a couple different options. One of them is the anterior lateral line. So you can differentiate a black carp and a grass carp based on the shape of the anterior lateral line. And this is from the initial description of both species, in that a grass carp coming off of the operculum, it takes a dip towards the pectoral fin ray and then continues straight along the length of the body. On a black carp, as the scales, those lateral line scales leave from the operculum, they're nearly straight, take a dip around the dorsal fin, and then continue nearly straight along the length of the body. Lateral head morphology is based on observations and reports from commercial fishers that initially collected black carp in the Atchafalaya River system and reported that to NICO. They said that black carp had a more pointed head or snout than grass carp, and that's how they were able to identify them. And you can see that here. We applied a truss network analysis with different points from the images that we had of wild caught fish. And the red arrow is the measurement for grass carp that shows that they had a deeper head at the preoperculum than black carp. And black carp had a greater distance between the landmarks at the eye and the preoperculum, that yellow arrow there. So you can see that they do have a little bit more pointed head than grass carp, and that's based on the measurements in those regions. Also, from a history of working with these species in culture and from various just prior research, we found that the visible presence of the premaxilla when viewed dorsally with the mouth fully closed was a good indicator. So on a grass carp, you can see in this image here, I have an arrow pointing it out that that premaxilla is very visible, that line. A lot of the images I had from my work are much darker, and I was worried that you might not be able to see it from the contrast on the slides that I presented here. So I have this image from the Fish and Wildlife Service, and I believe that's a tank fish. So you can see fairly well with the lighting and everything else, that premaxilla. These characters, we saw some plasticity in them, and obviously with lateral line scales, scales in particular, those fish can lose a scale. So we recommend a combination of this character set when trying to identify fish. And the character set and the outreach materials that have been used to create it are available on invasivecarp.us. I actually have the link to the PDF on the bottom of it right there that you can see if you're interested in that. So moving on to distribution, all these points are from our U.S. Geological Survey Non-Indigenous Aquatic Species Database. And I've created this map for you. You can see that the first record we have was from 1994 from that NAS database, and that is an aquaculture escape, but there's actually a pretty heavy history of use in Arkansas, Mississippi, and Louisiana. There just aren't any documented escapes from those locations. The first reported capture in the wild was from 2003, and I have an arrow pointing there. This is Oxbow Lake at the Middle Mississippi River. And then again, I mentioned on the previous slide that Nico's prior work with commercial fishers in the 1990s. Those folks were down in the Atchafalaya and lower Red Rivers, and they were some that mentioned undocumented commercial collections of black carp from those systems. I should also note that when I put together this map for you, it has kind of a timeline of 1999. So I want to point this out, 1994 all the way up to 2019, because on a later slide, I plan to highlight some of the more recent records, but a substantial number of these come from 2017, 2018 timeline, which I'll mention in a second. But black carp reporting was somewhat sporadic until 2015. That was the start of a program by the state of Illinois, bounty program for captures that were recorded in Illinois or adjacent states. And you can see how that's expressed by the density of points around the state of Illinois here. During that time frame, we worked directly with commercial fisheries markets or commercial fishers down in the lower Mississippi. As I mentioned, again, lower Mississippi is this extent down here. We have the Atchafalaya River and the Red River coming in here. And that was in 2017, 2018. Our goal there was just to provide some base samples for the life history research that we were trying to do with the within the previously described species range. So a recent change to that Illinois program beginning in 2021, They expanded it a little bit, so it now encompasses, and this was the outreach document that I showed you before, but the Missouri, Upper Mississippi, Ohio, Cumberland, Tennessee river systems. And there's also continued work within the waters of the state of Illinois where it has been. And, with this, we kind of anticipate that data will continue to be gathered on species distribution and be able to continue ongoing life history research within the waters where consistent capture has occurred. And in the past, the commercial, most of the records that we get come from commercial fisheries. We've had some recreational fisheries and reporting, but typically it tends to come from commercial fisheries, and kind of anticipate with this expansion, we should be hearing from more of those commercial fisheries. And this has been a good tool for monitoring the species range and range expansions within this basin. Again, I wanted to highlight that the upper star here is confluence of the Missouri River. So upstream of that is the upper Mississippi. Middle Mississippi is that stretch that goes down to the confluence with the Ohio at this star here. And then downstream, we have lower Mississippi. So just in recent years, we've seen continued captures from waters of the state of Illinois within the Illinois River system, which is right here. And that is the connection through to Chicago and the Great Lakes. We've seen extent change to kilometer 310, and the Ohio River, 552 kilometers upstream. You can see the red points here with some recent range expansion from the initial observations in Indiana. The upper Mississippi River, we've kind of seen the extent near the vicinity of Keokuk, Iowa. Pool 20 hasn't changed much in the past few years. The Missouri River, most of these points, I mentioned that recreational capture here, most of these points are based on the effort of a single commercial fisher that's filled in some of those locations at that spot and continues to report. And then I wanted to mention again, that timeline 2017, 2018, you won't see any red or green dots from the lower Mississippi River or any of these systems because we have not worked down there since that time frame. So it's not that the fish are not present within those systems or being collected. It's just that there isn't an incentive for people to report, and they don't report. So we know that they're still there. We just haven't had any reports that fill in the gaps between those clusters. And then you probably noticed looking at this map, this isn't an error, a GIS error. This is actually a single record from West Virginia and a pay pond system where the state identified black carp, and we confirmed it based on an image, and we haven't had any more reports from that system or beyond that Mississippi River basin. So checking back in with our timeline, I went all the way through distribution. You got an idea of background. So now we're kind of transitioning to what do we do as far as life history research with the samples that are retained from the wild caught fish, and then what are the specific laboratory and field research projects that we have ongoing. So as far as population parameters, this is a lot of what we learned from life history research. We've had samples collected since 2009, even prior to that 2015 increase in samples. These have come from the wild and been processed for life history research, and that consists of determination of origin, population genetics, reproductive condition, aging, growth, and diet. And I'm going to follow a format on the next few slides. If it's in bold here, these are topics that I plan to cover on the following slide in a little bit more detail. If it's not in bold, then I'll just briefly highlight that on the slide that it's on. So I did want to note, so within the population genetics work, Rick Lance is working on an update to population level genetics from the samples that have been collected from the wild. Till recent times, there was an initial population genetics study by Hunter and Nico. If you're interested in that topic and need to reference it. So this slide is basically a combination of all the information that was on the past one. So there's a manuscript that's in review that looks at origin and status of black carp in the wild. Basically, it's by Greg Whitledge at Southern Illinois University, and its establishment of invasive black carp in the Mississippi River Basin, identifying sources and year classes contributing to recruitment. So this takes the otolith microchemistry from SIU, ploidy analysis from the Fish and Wildlife Service, reproductive development in the form of gonadosomatic index, and age estimates through 2018 through us at CERC. And we combine that information to look at those sources and year classes contributing to recruitment. So basically some of the conclusions from that: wild black carp primarily consists of fertile, naturally reproduced fish, so non-triploid fish. We do see some introduced or escaped fish still present, but there's minimal detections of those fish, either from the otolith microchemistry or from floating analysis. We've seen that reproduction has occurred in several rivers, multiple year classes from the otolith microchemistry, multiple year classes of wild fish are present, and wild fish have recruited to adulthood. Okay. Moving on to ecological effects. Next, I have diet. I will cover the diet research that we've done with large juveniles and adults on the next slide. Also know that one of Greg Whitledge's students, kind of in parallel to this, was studying the isotopic niche of black carp. We provided tissue samples for that from the wild carcasses, so we kind of looked at the diet of fish, the physical diet of fish at the same time as he was studying the isotopic niche. And Hudman actually found that they have a fairly broad niche based on his work, and it's similar to what we found from our diet work, our physical diet work as well. There's also another manuscript I wanted to bring up, some recent work on young of year and juvenile feeding behavior done by the Illinois Natural History Survey, and they looked at the differential vulnerability of native and non-native mollusks, particularly bivalves, to predation of juvenile black carp. And I mentioned I wouldn't go into detail on it, but there has been some food web modeling done by the folks at NOAA. And then again, this is a topic area when the binational risk assessment presentation is given at a later date. If you're interested, please follow through. So as far as diet, I'm going to follow a format here where I have the citation to the manuscript in the upper right-hand corner. So this is our first manuscript on diet, our first examination of diet items consumed by wild-caught black carp. And sample size here was 109 black carp collected from 2009 to 2017. And some of the things that we found from this: black carp predominantly consumed benthic prey. Native mussels and snails were present in the diet. Corbicula and dracidians were present, but at a low percent incidence. And then we found that they also consumed insects in the form of mayflies, midges, and caddisflies. There was some evidence of flexibility in feeding modes. There's an example that I give here of the subimago stage of mayflies, and those are pictured here in this collage of diet items. There were other taxa that were also consumed that suggest, mid-water column subsurface feeding. And then we also saw local consumption of numerous items of a single taxa. So some of these samples of the gastrointestinal tract of black carp had numerous diet items. Snails is the example that I have in the image here. We also had chironomids as well, and a few other taxa. If you're interested, please follow through and look at the reference for more details. We had a recent manuscript that came out. It pertains to the presence of this fluke. Fluke pictured here, Spedigaster contricola, and its presence in the gastrointestinal tract of black carp. This is from the same pool of samples with some additional samples added over time. Basically, the way we've studied diet is through these commercial captures. And with commercial capture, you're dependent on the commercial fisher's gear to understand what that fish consumed. And with that, in particular, I'll get to this on a later slide, but hoop nets are a common method for collection. And with hoop nets, they may be fished for 48 hours to 72 hours, depending on the market. There's some variation in, methods that they fish, how they fish, where they set. But that gives the fish the opportunity to enter a net. If it's at the start, it has time to evacuate its gastrointestinal tract, and it may not have any contents there. Another thing that we learned from tank studies on the species is that they may not necessarily consume every piece of a diet item that they ingest. And I'm referencing particularly bivalves or snails that they may be able to, we've seen with corbicula that in tank studies that they'll engulf the clam, break the clam with their pharyngeal teeth, and they can reject pieces of that shell. So they aren't necessarily ingesting all of it. And you're familiar with mussel identification, at times you need to be able to have certain pieces of the shell to identify a mussel down species. So with all of that, we had some or a numerous amount of samples that didn't have anything present in them other than these flukes. And the flukes are a mollusk parasite. So they can be used as an indicator of prior mollusk consumption. And with that, we were able to look back and understand that the estimates and the indicator of the importance of mollusks to the black carp diet may have been an underestimate from the initial study. Also, as you can see in this image, there was some implications for parasite dispersal because of these flukes within the gastrointestinal tract and the presence of gravid females. So with this, we've transitioned research to kind of focus on the mollusk component a little bit more to understand the taxa that are being consumed and the habitats that they may potentially occupy or where they're coming from and what type of mollusks are being ingested. So we focus a little bit more on the presence of those. I do have here on the right-hand side the list of species that have been identified. A lot of our mussel work in particular with the diet is again based on physical identification and the requirement for the presence of the shell fragments that can be used to aid identification. So with that, a lot of times we do have to leave it at union a day and cannot identify beyond that. I should mention also that with that expanded range, we do anticipate obviously a greater potential list of species that could be understood from the range of reports and potential range of mollusks and black carp as those reports come in. So transitioning here to life cycle and some of the other points, I wanted to give you this background. So again, from Nico et al., a great reference, reproduction occurs with an upstream migration in the main stem, spawning in free-flowing waters. Egg development occurs with downstream drift. Spawning is in late spring and summer, and it's stimulated by a rise in water level and water temperature. Larvae migrate to backwater areas where they develop, and the young and juveniles develop in these areas as well. And then the main stem is used for feeding and wintering habitat by subadults and adults. So that all correlates with detection, understanding the distribution of the fish and the size of the fish. The two topics I was going to cover here are environmental DNA and physical capture. Environmental DNA, and I won't expand on this too much, but obviously it's a tool being used by a lot of different subject areas to understand detection of different organisms, and it's been used for black carp as well. It's still being developed though. Rick Lance and at U.S. Army Corps of Engineers at URDIC and Kathy Richter at my lab here have been studying it. Rick has published on some assays for black carp in particular, and they've continued to do their field work and lab work pertaining to eDNA development on the species. As far as physical capture, there were some early pond studies on different methods for collection of black carp, electrofishing and fight netting in particular. There wasn't much success by those two methods in the field at this point, at the stages that they've focused on or the sizes that they've focused on. I also wanted to mention that based on an increase in commercial collections from a specific oxbow lake in the Mississippi River, there was one management action that was enacted. This report here that I have, the Horseshoe Lake Incident Response in 2018, that did successfully collect black carp by several different years within that effort. And then from our work with commercial fishers, we put together a summary of their reported capture methods, the specific gear dimensions, and I'll get into that in a later slide. With that, though, I did want to mention that our colleagues at the Illinois Natural History Survey are doing some work of their own on capture on the use of baited versus non-baited hoop nets, and then also enhanced detection of black carp in the lower Illinois River, which is an area of interest. And then on later slides, I will get into the collection and identification of early life stages and what's been done with that. So there's minimal information available from the species native or other introduced ranges. Again, I have the same format here. You can see the reference in the upper right-hand corner to the summary that we put together. This is basically a report of the capture data with different gear dimensions that came from the commercial fishers that successfully collected and reported black carp to us. So our objectives with this were to assess the course habitats, time of year, and methods of collection. The time frame is 2011 to very early in 2019. We looked at 302 records with specific year dimensions. Size range of the fish is all the way from 400 to that largest 400 millimeters total length to the largest fish that I mentioned, the 1607 millimeter total length fish. But know that most of the records that we received during this time frame were the around the 700 to 1,000 millimeter total length size. And from this, we found that several gears collected black carp, specifically hoop nets and gill nets, and those were fished among main stem habitats and backwaters throughout the year. There is some seasonality to how those gears are fished, and that's represented also in the time of year that they successfully collected black carp. Hoop nets set in May, June, and July were kind of the highest reported by that gear. And then gill net catch was highest in November, January, and February. And that matches a pattern of effort where commercial fishers will more commonly fish hoop nets during the spring, summer months, warmer water temperatures, more active fish than in the winter when they tend to fish gill nets. Please note with this that data are dependent on commercial fishing effort and the catchability of gears. One thing, in some of the states in the U.S., commercial harvest is restricted in tributaries. They're kind of set to focus on their efforts in the main stem, larger systems. And collection and those records are dependent on the distribution of effort. And like I mentioned, a lot of our reports come in from commercial fishers. In the past 2 years, I've seen a few more reports by hook and line anglers, but not many. Most still are commercial collections. So that said, tributaries are an area where we could learn a little bit more just based on that lack of effort and information that comes in. And I wanted to point out one thing when I say gill nets, there is the use commonly of this tied down gill net, which is a gill net set to a slightly shallower depth than it would fish if it was fully stretched. And that creates a little bit of a bag. It doesn't act fully like a trammel net, but it creates a little bit of bag and leads to a little bit of enhanced capture according to the commercial fishers. And they commonly use that for several species. So with that information, we've moved to target black carp on our own. We use hoop nets, as I mentioned, in May, June, and July, and we've done that in 2020 and 2021. And our goal with this research, I don't have any early results to report. We have successfully collected black carp. We've focused on an area of increased capture at the confluence Missouri and Mississippi River systems. And we're trying to understand the environmental covariates that lead to capture, such as depth, current velocity, river stage, and water temperature that facilitate that capture. And some of the point of our efforts are to also facilitate telemetry research through the acquisition of fish and the surgical implant of transmitters for that work. So on this slide, I have our telemetry research, which we're just trying to understand the habitat use and movement of fish. Basically, I mentioned how a lot of what we understand is dependent on that commercial fishery, commercial effort, capture, and tagged fish provide information about habitat use and behavior independent of the fish, how different gears fish and how someone goes out and fishes. So also independent of catchability. So again, we're focusing most of that initial capture and tagging effort at the confluence of Missouri and Mississippi rivers. And that's an area of increased commercial capture. Majority of our data have been collected by active tracking. And the distribution of our telemetry effort has changed and been dependent on fish movement. We initially kind of started around this confluence region, as you can see by the dense points, but we've expanded our range downstream, upstream in several systems to be able to gather that data based on the behavior of our fish. So we do work with several partners on this, the Corps of Engineers, other colleagues at USGS, the state of Missouri, Missouri Department of Conservation, and they manage receiver arrays as well as us. I don't have those plotted on this map here, but we do work together on a fairly large effort and assist with other telemetry studies as well. Just some initial observations. We've seen some intermittent use of tributaries by individuals. We've seen some site fidelity by individuals relative to main stem habitats. They may be tagged in an area, they tend to occupy it. They may disperse for a brief time to another habitat and then return back, or another area several miles away, and then return back to that same area and occupy it for a fairly consistent time frame. We've seen dispersal that's occurred upstream in the Missouri River, as I have pictured here. I mentioned the tributaries. This is the blue circle is the Osage River. Again, we've seen use by one fish and intermittent use by another. We've seen dispersal upstream through one dam during open river conditions. I should mention that the Mississippi and Missouri rivers are both exhibit seasonal changes in hydrograph and flood events. And based on that, we say open river conditions are when the different gates on that dam are fully open. So it presents more of at high water, more of an open river, more accessible condition. And that's when we saw dispersal upstream in the upper Mississippi. So with this study, we'll get a better understanding of habitat use, obviously, also some of the post-tagging behavior and movement that we anticipate will assist with potential future seasonal research. And then also understanding of the general broad scale movement patterns of the fish. So onto the early life stages, I've kind of focused on the large juvenile adults throughout most of this, and that's because we don't have a whole lot of reports of the early life stages. Very little reporting as far as collection records. No eggs or larvae have been collected. I know ichthyoplankton surveys are fairly consistently run in the Illinois River by the Illinois Natural History Survey. In Louisiana, there's been a several year-long study ongoing in the Atchafalaya, Mississippi, Ochita, and Red Rivers, and they haven't detected black carp. As far as age 0 fish, so a slightly larger stage, and I have some pictured here on the right in this slide. We did see consistent reports from 2015 to 2018 by Wes Sleeper for the Missouri Department of Conservation during his tenure with the agency. That's the upper image on the right there is kind of the size class that he targeted. It was late summer typically. And then we have a single collection from Gar Creek in the Ohio River in Kentucky in 2018 by Kentucky biologists. Both of those are floodplain habitats of large rivers in the Mississippi River Basin. They're actually fairly close to each other. I wanted to provide for you, jumped ahead one. Just an idea where these are. You can see Dutchtown Ditch is basically kind of an agricultural ditch adjacent to the floodplain. This is the Mississippi River here. This is the Ohio River here. These are at different scales. By the way, just screen captured. This is Gar Creek. And you can see that this is the confluence of the Mississippi and Ohio rivers. They're somewhat in close proximity to each other. So let's give you an idea. These are, again, floodplain habitats of both of those systems. We also have a record from this diversion channel here as well. So this is certainly a life stage that we can study more and understand better. I just completed some sampling with the Fish and Wildlife Service on a lab study to potentially use electrofishing for inducing capture-prone response of juvenile black carp from the 70 millimeter to 500 millimeter total length size class. And we see that may be a potential method for sampling these floodplain tributaries of large rivers. Also, biologists in Kentucky within the next year will be attempting to sample the lower Ohio River to collect juvenile black carp as well. So again, we've not seen eggs or larvae collected. We're working to develop methods to study early life stages though. One of the things that we need to understand better is characterization of black carp egg and larvae development, and it's necessary to inform drift models to better understand recruitment dynamics and improve identification and validation of larval black carp where recruitment does occur. We've been a little bit limited by this due to the lack of availability in aquaculture to acquire broodstock fish. I mentioned before that black carp mature at large sizes. Females, you're looking at about 1 meter total length. That's a fairly large fish for aquaculture operations. And late ages, from the aquaculturists that we've spoken to, we know that 6 to 7 years is about the timeline for a fish to mature in the ponds. I've been working with several states on this to try and acquire broodstock for spawning and development of this research in our biosecure facility at our lab. We've worked with commercial fishers to get the fish and states to permit through that. This is an example of the flu egg model here, just to give you an idea of the drift model that data could be incorporated into. So moving on to controls, I plan to talk a little bit more on the next slide about toxic bait development and the work that I've done. Just know that other technologies in development by USGS have not been studied on this species in particular. You know, within the existing deterrent and attractant technology, the one that I'm aware of as far as acoustic deterrents, there's a thesis that I have listed here that I believe Mary Beth Bray helped on, but black carp are included in the hearing thresholds and impact of anthropogenic noise, but that's about the extent. You know, other deterrent and attractant technology consists of, again, acoustics, carbon dioxide, electric barriers. The bioacoustic fish fence, if you're not familiar, is a combination of an air bubble curtain, acoustic stimuli, and light as a deterrent. And then there's also the development of the oblique bubble screens. So for the bait development work that we've done, the goal was to develop a single dose piscicide species specific bait for black carp. So there's kind of two options for pesticides at this point, Annamycin A or rotenone. Annamycin A was chosen for its toxicity at lower doses than rotenone in early testing. We anticipated, if successful, that the application for this would be for protection of specific endangered mussel beds, so kind of a targeted effort and not necessarily the broad spectrum control that you might think of with other pesticide applications. And we chose a certain design for this. We have not been successful at producing that single dose species specific bait. We did develop a few things. We developed a delivery method that consists of a live mollusk attractant. You can see that picture here with the curricula. And this is the fully assembled, bait here. Anticipated that a fish would see the search image of that live mollusk. You know, the live mollusk feeds, excretes, kind of represents the scent, the search image of a diet item, and hopefully would be interacted with more regularly than maybe a food-based product. Also, taking this approach over alternatives, the pesticide is retained in a glass ampule because Annamycin A degrades by hydrolysis. So with a low density black carp population, we anticipated we needed that ampule to be able to have a treatment out for a longer amount of time to interact with our target species. We also did some forage size preference testing with the curricula, and we looked at ingested toxicity of Annamycin A to black carp and grass carp. The results of the grass carp work are actually being implemented in development of baits for that species at this point. And with that, I'm back to kind of my summary and my outline, and I just hope that you were able to take something useful about the black carp in general, their status in the United States, and this summary of research. And with that, I wanted to thank our funding sources, the Great Lakes Restoration Initiative, U.S. Geological Survey, Illinois Department of Natural Resources assistance with collecting black carp, and then these numerous agencies that have assisted throughout with all of this work. And with that, I'll take any questions.
Thanks so much, Patrick. Really enjoyed your presentation. Particularly enjoyed when you had the teeth bones. That was really great to see. Nice to get a visual and amazing the size of them. So that was great. We do have a few questions that have come in, but please feel free to add any more if you think of any as we're speaking. But the first one that came in, somebody is wondering what the closest capture to the Great Lakes, Michigan, and how far from the Great Lakes is the closest known reproducing population of black carp.
Let me see if I can bump back a slide for you. Probably easier to show than to tell. So,
for sure.
So on this slide, I mentioned the Illinois River and the extent of lake in Illinois. So this is 310 kilometers upstream from the confluence of the Mississippi. I believe, and I had to measure this myself, the distance may be off, but I believe it's a little over 200 kilometers from the Great Lakes, that point.
Cool. Yeah, that's I guess on our side, not the best news, but but yeah, thank you. In terms of the next question, we've got somebody had said that they read a recent newspaper article citing that a merchant in Michigan area, perhaps they're not sure, was fined about $15,000 for selling invasive carp meat. They're wondering if there's any interest to aggressively harvest invasive carp for such uses, and which would then also act as a control mechanism. Do you have any thoughts on that?
I don't quite understand the question. There are markets that exist for silver carp, and there's a decent amount of commercial fishing and various programs to use commercial fishing as a control of silver carp. At this point, we don't see a lot of black carp reported enough to have a market. That Illinois program is kind of the closest thing that approaches that, where it's getting the word out there about the presence of the species, and it promotes a method to harvest and report.
Interesting. Yeah, thank you.
Mm-hmm.
I'll go in chronological order if that works.
Okay,
So we might be jumping around a bit on the topics. Somebody was wondering if they might have missed it, but are there only sterile fish being tagged and released?
Only feral fish?
Sterile.
Sterile fish? No, we use wild fish. They're not sterile. So we don't introduce triploid fish for that work.
Okay. Next, is there any evidence of black carp consuming faucet snails?
Faucet snails? I don't believe we've seen that in any of our samples, and I'm not familiar with faucet snails from any prior research.
Okay.
If they want to follow up and ask, I'm going to bump this back here. It's my last slide. There's my email address too. If somebody has a specific question about past work or anything similar to that, they're welcome to email, and I can follow up as well.
Sure. That's fantastic. Yeah, thank you for adding that. And likewise on our end as well, Invasive Species Center, we can try to respond to anything as well. So the next question, somebody said the excellent talk. So is tagging and releasing black carp contributing to their spread and impacts? Is there not enough published studies documenting their biology and impacts to justify removing all captured black carp to prevent their movements towards an introduction to the Great Lakes? So a few parts to that one.
Yeah, there really isn't a lot of information from the species native range to be able to understand the distribution of the fish. And with what we can learn from tagging a few individuals, we can understand better how to develop control methods and how to collect fish to have an impact. And yeah, in the area that we have focused our work is an area where we see increased reports in commercial captures in general. So we know that there's fish there. We just don't understand fully how to collect them, how to control them, and potential impacts, because it's so dependent on those commercial fisheries, which within a reach may be only a few fishers, and their range may vary seasonally based on whatever market they're targeting. A lot of the markets we see are either buffalo, catfish. There's some grass carp, some silver carp, but a lot of it is the native fish and small kind of local markets and effort.
Interesting. I guess kind of related to that, and related to the question before on specific type of snails. If they invade the Great Lakes, will they eat zebra mussels? And have you involved another part to this question, but have you involved First Nations with this program, since they are unable to fish in length using gill nets, and they could be a great aid to the program, this person's written. So, sorry, two-parter again.
That's okay. For your first part about zebra mussels, that's a pretty common question, and that's why I put it in there. With that initial study on diet, we did see that zebra mussels are present in the diet. But it's a low percent incidence among the samples. We do continue. So with the transition of diet research, we are focusing more on mollusks. So any sample that contains shell fragments, we continue to retain those for identification. And we're going to look at if that percent incidence changes over time with the samples that we have or within a specific reach or area. The goal there is to understand you know, potentially impacted unionid mussels, but also that invasive component as well. So they do consume zebra mussels, but it's not, they aren't specifically targeting them, if that. And then to the question about First Nations and their fisheries, it sounds like within the region that you're talking about, they have some special regulations and ability to go out and target fish, and that's a little bit different than the area where we've kind of focused, and that's more of a management action as well. I don't, we just try to study based on, you know, what actions are being taken by the states. And as far as the control component, you know, we kind of developed the controls. Have not, and most of what I've done has been related to working with those commercial fishers and existing markets and fisheries.
For sure, yeah, thank you. The same person has also asked, just in terms of feeding, are black carp able to be used into supplemental usage like livestock feed, dog and cat feed? Is that something that's been explored?
So again, the species is not, it's not really collected in the density that you would need to have a market like that at this point.
Okay. Yeah,
but it has been studied as a food fish within the native range and other introduced ranges. Sorry.
Great. Yeah, yeah, because I have heard that before with grass carp, I think, but yeah, that's interesting with black carp. It differs a little bit. So do black carp have any close native analogs similar to, with species with very similar niches that they're thought to be directly competing with? Is that something you've looked into?
Yeah, so we're not really at the point to be able to say that. You know, that's kind of where the diet research falls, is just trying to understand what niche they fill. With Hudman's thesis, they looked at, I believe, blue catfish, freshwater drum. They thought that there would be kind of an overlap there. And the freshwater drum and the blue catfish, I think, ordinated separately. They had kind of their own different sections. And then if you look at the ellipse for black carp, it was a much larger ellipse that coincides with what I was saying is a broader niche. And that's kind of what we saw from the physical diet as well. They have that fairly broad niche.
Interesting. Yeah, we're just getting a couple comments in as well, based on some of our earlier conversation on black carp being rare, rare captures. But maybe we'll stick to maybe one more question just in the interest of time. Sure. So somebody has asked, if their maturation is so late, shouldn't it be reasonable to think that they could be over harvested easily? I guess that kind of goes against what we just mentioned. Yeah.
Yeah, they do mature at a fairly late and large size. It's just understanding how to collect the fish is difficult. That's kind of the stage that we're at.
For sure. For sure. Well, thank you so much. We really appreciate a lot of questions there. So thank you so much to the audience for participation. And if we didn't get to your question, or if you have any further questions, as Patrick mentioned, his contact information is on the screen, fortunately. And ours is available on our website. And so, yeah, with that, just would like to say again, thank you for presenting. This webinar will be recorded and posted on our website at asiancarpcanada.ca and the invasive species center.ca as well. So just a reminder to also take the survey at the end of this webinar as well. If you could do that, we would really appreciate it. So thanks so much again, and take care.
Thank you.